Hello all, welcome to KU Padasala. Today we will discuss the role of metabolism in cancer. Okay, I am Shankar Babu. I come from the University College Trivandrum. I teach botany there, and today we will learn what is the role of metabolism in cancer. So first we will see what are the hallmarks of cancer. So cancer cells they divide uncontrollably because they resist cell death or apoptosis, and they have constant proliferative signals that is coming from the intrauterine epithelial cells, and they evade growth suppressors, and also they activate the invasion and metastasis. Then they are immortal. So there is something called replicative immortality. So these are some of the aspects that help the cancer cells survive. And when it comes to solid cancers, there is also angiogenesis, which is called invasive angiogenesis, and these are the factors that help them grow uncontrollably. So when you see the hallmarks of cancer, there are certain new hallmarks that have been recently discovered. The first one is the evasion of the immune system. So immune system attack by the immune system is evaded by the cancer cells through this methodology, and then there is deregulation of cellular energetics. So that is what we will learn today. So how the cancer cells alter their metabolism in order to sustain growth. So if you see the metastasis of cancer cells, from the site of origin, these cancer cells move to different parts of the body. So common examples is the cancer of the lung will spread to the brain, or the cancer of the kidney will spread to the adrenal glands. So this process is known as metastasis or distant metastasis, because from the site of origin the cancer cells migrates to different parts of the body, and this is particularly through fluids, blood, as well as the lymphatic system. Okay, so initially the cancer cells that are growing attached to the media, they will slowly migrate from their site of origin by the secretion of certain enzymes which are called matrix metalloproteases. Okay, so these proteases will digest the tissue membrane, and the cells will slowly migrate to the liquids, that is either the blood or the lymphatic system. Once they reach the lymphatic system, they are free to move about the different organs within the body, and once they find a suitable site for invasion, they will settle down there, and this particular phenomenon is called distant metastasis. So initially the cell has to remove from the substratum, and they will slowly move to the next site, and there they will have to do attachment. So there is a differential expression of genes that will help them do this process. So finally, after the first wave of metastasis, another group of cells will migrate, and this produces the second wave of metastasis, and this will continue until it spreads throughout the body of the organism. And in order to help the cancer cells achieve this phenomenon, there are certain pathways that are activated. So some of the few pathways that we can think about is the viability circuits. You can say they are the viability circuits. So wherein these cells will improve the motility, and they will slowly move to the distant sites. So what are the different types of treatments that are available currently? So you can think about antibody therapies, telomerase inhibitors, anti-inflammatory drugs, inhibitors of growth factors, inhibitors of vascular endothelial growth factors, inhibitors of PARP, and inhibitors of glycolysis. So what is glycolysis? That is what we will learn today. Before that, we have to see what causes the cancer cells to move from its site of origin to different sites. So one of the major factors that help the cancer cells metastasize is the hypoxia. So what is hypoxia? As the word suggests, it is the lack of oxygen. So what is the percentage of hypoxia that the cancer cells feel? So a percentage of oxygen which is less than 5% is considered as hypoxic condition. So okay, so under normal conditions, normal oxygen conditions is termed as normoxic conditions or normoxia. Hypoxic condition is less than 5% of oxygen in the tissue system. So under normal conditions, there is an enzyme that is called prolyl hydroxylase. This prolyl hydroxylase will phosphorylate a protein. This particular protein is called HIF1 alpha, hypoxia inducible factor alpha. So once this prolyl hydroxylase phosphorylates this protein, it will get degraded. by the ubiquitin system of degradation. So, this HIF1 alpha is kept under check under normoxic conditions by this phosphorylation. So, under what hypoxic conditions, what happens is that this enzyme is deactivated. So, once this enzyme is not available, the phosphorylation of HIF1 alpha will not take place, and there is the stabilized production of this protein, and this protein in turn will induce the metastatic genes within the cancer cells, and they will help the cancer cells migrate to distant sites. So, this is how hypoxia helps the tumor evade the immune system as well as move to different sites of the tissues. So, the HIF1 alpha expression will increase with the hypoxia. So, as the tumor moves away from the size of the tumor increases, the cell that is present in the center of the tumor is away from the blood vessel. So, blood is what brings the oxygen to the tissues. So, once the center of the tumor experiences hypoxia, they will start expressing the transcription factor, which is the HIF1 alpha. Once HIF1 alpha increases, it will produce genes that help them evade the immune system and migrate to distant sites. So, this is the picture that shows the hypoxic cells and their movement from the site of origin to the distant metastatic sites, and there are certain processes that is involved, that is the degradation of extracellular matrix and matrix remodeling. All these are helped by the hypoxia inducible factor, transcription factor, that will be produced. So, this is a cartoon that will represent the movement of the cancer cells under hypoxic conditions, and every gene that is expressed during hypoxia is under the control of the transcription factor HIF1 alpha, and HIF responsive element HRE is the transcription binding site for the HIF1 alpha. Once the HIF1 alpha binds to this transcription site, it will produce the production of genes as well as proteins. They will help the cells migrate from their site to the other. So, metabolism is increased glycolysis. So, when you say glycolysis, it utilizes glucose and it produces pyruvate. So, once pyruvate is produced, it will enter the mitochondria, it will undergo aerobic glycolysis, and it will enter the Krebs cycle or the TCA cycle, and finally the electron transport chain that produces the ATP molecules for the energy. So, if you look at the cartoon, you have glycolysis, Krebs cycle as well as oxidative phosphorylation. So, glycolysis takes place in the cytoplasm, Krebs cycle or the TCA cycle takes place within the mitochondria in the mitochondrial matrix, and oxidative phosphorylation takes place in the electron transport chain that is present in the inner mitochondrial membrane. So, if you look at the production of ATP, glycolysis induces the production of 2 molecules of ATP and 2 molecules of NADH per molecule of glucose utilized. So, in Krebs cycle, this amount of energy that is produced in terms of ATP as NADH is 2 ATP, 8 molecules of NADPH and 2 molecules of FADH2 per glucose molecule, and coming to oxidative phosphorylation, it comes to 3 molecules of ATP and 2 molecules of ATP, that is 3 molecules of ATP per NADH molecule that is utilized, as well as 2 molecules of ATP per FADH2 molecule that is produced. So, in total around 32 ATP molecules are generated by glycolysis, Krebs cycle as well as oxidative phosphorylation together. So, this is the energetics in a normal cell. So, what happens to the machinery in a cancer cell? So, we should understand that in cancer cells there is a lack of oxygen or hypoxic condition exists. So, what happens is that the cells will utilize glycolytic pathway to generate ATP, as well as it will shuttle many of the glycolytic intermediates to synthesize nucleotides, proteins as well as macromolecules. So, as you all know, cancer cells have to continuously divide. So, in order for them to divide, they need DNA, they need proteins, they need RNA. So, all these molecules which are large sized molecules or macromolecules have to be synthesized from the glucose that is utilized by the cells. So, in order to achieve this raw materials for the production of these macromolecules, many of the intermediates of glycolytic cycle is shuttled to produce these macromolecules, including nucleotides, amino acids as well as carbohydrates. So, this is what happens in a cancer cell. So, in a normal cell where most of the glucose is shuttled to the mitochondria in the form of pyruvate, and then pyruvate is converted to phosphoenol pyruvic acid, and then it moves inside the mitochondria. In a cancer cell, what happens is that this pyruvate is immediately converted to lactate, and this conversion of pyruvate to lactate is happening by the enzyme lactate dehydrogenase. This is a reversible enzyme, so pyruvate will be converted to lactate and lactate can be converted back to pyruvate. And the production of lactate becomes very important for the cancer cells, because in a solid tumor the secretion of lactic acid or lactate produces an acidic environment. So when the tumor is present in an acidic environment, the immune cells will not be able to target these cells and they are protected from the immune system. So this is the major phenomenon by which cancer cells get addicted to glycolysis. So as I already mentioned, 2 molecules of ATP are generated during the glycolytic cycle, but more than energy, the macromolecule synthesis, the raw materials for macromolecule synthesis is shuttled through glycolysis. And in cancer cells, the glycolysis is active also during normal oxygen conditions, that is normoxic conditions. So this particular phenomenon is called aerobic glycolysis, or it is also called Warburg effect, after the scientist Otto Warburg, who discovered this phenomenon in cancer cells. So basically cancer cells will prefer to use glucose and convert it to lactate, and this particular phenomenon is called aerobic glycolysis, and approximately 4 moles of ATP will be produced per molecule of glucose during this phenomenon. So in order for us to target a cancer cell, what is that we have to do? We have to target this glycolytic pathway. So that is what we are trying to do through metabolic alteration. So why is the cancer cells addicted to glycolysis? So first of all, I mentioned the tumor microenvironment. So around the tumor, lactic acid secretion produces an acidic environment, and this acidic environment prevents the attack of immune cells in the cancer cells. Secondly, lactate acts as a signaling molecule and it promotes the growth in cancer cells. And finally, biosynthesis of macromolecules is helped by the aerobic glycolysis. So these are the reasons why which cancer cells are addicted to the glycolytic pathway. And also another important factor that we have to remember is glycolysis produces drug resistance. So drug resistance is a phenomenon in cancer cells wherein the cancer cells evade the attack of chemosynthetic drugs that we give as anti-cancer therapeutic. So one of the mutations is given. So BRAF is a protein kinase that is present in melanoma cells. So a mutation wherein valine is converted to isoleucine, which is represented at the 600 amino acid in the BRAF protein. So this will happen, what happens is that this will produces a relapse of the cancer. So slowly the cancer cells will evade the therapy. So this is because of the phenomenon that is called metabolic plasticity, that is cancer cells can shuttle between aerobic glycolysis or Warburg effect and oxidative phosphorylation for their energy requirements. So glycolytic pathway that is given in red, and the oxidative phosphorylation is given in blue in this cartoon. So you can see that the oxidative phosphorylation is taking place within the mitochondria. So there are different ways in which the pathways are targeted by different molecules. So basically what we have to remember is that during the glycolytic pathway, different macromolecules will be synthesized from the intermediates of glycolysis, and that is what you have to see from this particular cartoon. And within the mitochondria you have the mitochondrial oxidative phosphorylation takes place. So as an example, we will take one enzyme, that is PKM2. So pyruvate kinase M2 is the abbreviation of PKM2, and it is a very important enzyme for cancer cells. So how is it important? So pyruvate kinase, as you know, is converting the phosphoenol pyruvate to pyruvate, so which is the penultimate step in the glycolytic process. So pyruvate is converted to lactate by lactate dehydrogenase, I already mentioned. So phosphoenol pyruvate conversion to pyruvate is taking place by the enzyme pyruvate kinase. So this pyruvate kinase has two isoforms, PKM1 or pyruvate kinase M1 and pyruvate kinase M2. So what we have to see is enzymatically PKM1 or the pyruvate kinase 1 is active. So whenever phosphoenol pyruvate is present, it will immediately convert phosphoenol pyruvate to pyruvate, whereas in the case of the M2 form or the mutant form, which is PKM2, it is converting this phosphoenol pyruvate to pyruvate in a very slow manner. So enzymatically this enzyme is a very slow enzyme. So what happens when there is accumulation of phosphoenol pyruvate? The penultimate steps of the glycolytic process will be slowed down. So intermediates will be available to be shuttled for macromolecular synthesis. So that is why cancer cells specifically express pyruvate kinase M2 isoform compared to the PKM1 isoform. And this happens because you can expect the pyruvate kinase enzyme to be present in either a monomeric form or a dimeric form or a tetrameric form. So this tetrameric form is more active, whereas the dimeric form is less active, and PKM2 prefers to be in the dimeric form. So from this cartoon you can see that the accumulation of intermediates of glycolysis will be shuttled through the pentose phosphate pathway for the synthesis of amino acids as well as nucleotides and as well as the lipids. So how does the production of PKM takes place? So as we all know, all genes in eukaryotes are produced by splicing. So in PKM there is an alternate splicing mechanism that is taking place, which produces two isoforms, PKM1 as well as PKM2. So PKM1 is produced by the joining of the exons 8, 9 and 11, as you can see from the cartoon, and PKM1 exists in the form of a tetrameric protein and it is active. But PKM2 is produced under oncogenic conditions. So whenever there is an expression of oncogenes are genes that are increasing the activity of the cellular division as well as they promote the occurrence of cancer. So these genes, for example, one example is c-MYC. So MYC is an oncogene, and whenever there is MYC overproduced in the cells, what will happen is the PKM2 isoform is produced, and this is by the production of heteronuclear RNP proteins. So these proteins will preferentially create the M2 isoform. The M2 isoform is produced by the joining of exons 8, 10 and 11. As I already mentioned, exon 8, 9 and 11, if they are joined together during alternative splicing, they will produce the PKM1 isoform, which is the normal isoform that is present in normal cells. Whereas PKM2 is produced under the expression of oncogenes like c-MYC, and whenever this exon is expressed, exons 8, 10 and 11 will be joined together and they will produce the PKM2 form, and which is existing as a dimer, and it is a slow enzyme. So this increases the propensity of the cancer cells to divide. So how is it different from the PKM1 and PKM2? How are they different? So they differ in the amino acid sequence, and 22 amino acids are different in both these isoforms, and PKM2 is enzymatically less active and it is expressed only in cancer cells. Other tissues in which PKM2 expressed is in fetal tissues, especially in the developing embryos. In all the other cells, PKM2 is completely absent and only PKM1 is produced. So we have an enzyme that is present only in cancer cells and it is not present in normal cells in an adult organism. So this is the enzyme that we can target, because we want to target the cancer cells that is in a specific manner, and we have the enzyme which is targetable, that is PKM2. So the only available molecule until now was benzoic acid derivative, and you require a very high dose of this molecule to target the cancer cells. So that is why we have to find alternatives for this PKM2 to be targetable. So we propose a new molecule, that is Lapacol. Lapacol we can try and use it to target the PKM2 isoform. So how we came to this molecule is, Lapacol is a molecule that is fetotoxic, that is it is toxic to fetal tissues. As we already know, fetal tissues express the PKM2 isoform that is expressed in cancer cells also. So we came to the conclusion that we can try Lapacol and target the PKM2 isoform of the enzyme. So we also asked ourselves the question whether Lapacol can be used to specifically target Warburg effect or aerobic glycolysis. So in order to achieve this, there is a new instrument that is present, that is called the Seahorse extracellular flux analyzer. So this extracellular flux analyzer is an instrument that will help us target or help us study glycolysis as well as oxidative phosphorylation in a cell. So normally if you have to study glycolysis and oxidative phosphorylation, you need to lyse the cells and analyze the amount of ATP, amount of lactic acid and the amount of ATP that is produced in a cell. So the amount of ATP is giving an indirect readout of the oxidative phosphorylation that is taking place within a cell, and the amount of lactic acid will give us the amount of glycolysis that the cell is undergoing. So two things are wrong here. The first thing is you are lysing the cell. So whenever you destroy a cell, the energetics and the kinetics of the reactions will be affected. So in order to prevent this, they have decided a new instrument which is called the extracellular flux analyzer. So here this instrument will help us measure the intracellular levels of ATP as well as lactic acid. So how does this instrument work? So two modes of this instrument can be used. The first mode is called the oxygen consumption ratio, or OCR. We can call it as OCAR, and the other one is extracellular acidification rate, or ECAR. Okay. So ECAR will give us a measure of the amount of acidity of the extracellular matrix, and this acidity is caused by the secretion of lactic acid. So lactic acid is produced by glycolysis, and this lactic acid is what is measured by the change in pH or acidity. So that is what is called ECAR, or extracellular acidification rate. OCAR is oxygen consumption rate. As you all know, the mitochondria needs to respire. So whenever the mitochondrial respiration takes place, oxygen is consumed. So the amount of oxygen that is used by the mitochondria is measured by this particular mode, that is OCAR. So both these modes are operable in the same instrument, which is the extracellular flux analyzer. So in this, the first step that we have to do during an OCAR assay is that we initially measure the basal respiration of the cells. So basal level, what is the amount of oxygen is consumed? So that is given in this diagram. So from this you can see the basal respiration is highlighted in blue color. So once the basal respiration is measured, you inject the molecule which is called oligomycin. Oligomycin is a drug that will inhibit the ATP synthase enzyme in the electron transport chain. So once oligomycin inhibits the ATP synthesis, your oxygen consumption level will fall. So this basal from the basal level of oxygen consumption, it further falls down to the base minimal level. Then you block the you destroy the inter and outer mitochondrial membrane flux of ions, which is the proton gradient. So once you destroy the proton gradient, there will be a rapid increase in the respiration level that is measured by the maximal respiration. Okay. And this drug is called FCCP. You can also use CCCP. Both these drugs will destroy the, it is an ionophore. So ionophore, what does they do? They will destroy the potential gradient between the outer as well as the inner mitochondrial membrane. So you have a proton gradient, that is gradient of protons or H plus ions that is extending between the mitochondrial matrix and the inner mitochondrial membrane. So if you destroy this proton gradient, what will happen is there is no electrochemical difference and no ATP synthesis will take place. So the oxygen consumption will be the maximum at this level. Then you finally give the drug either antimycin A or rotenone. Once you give this drug, what these drugs do is that they will block the electron transport chain 1. The complex 1 will be blocked by either rotenone or antimycin. So once you block the electron transport chain, the oxygen consumption will be reduced to the minimum. So this is the flow chart, and here you can see the different levels. So once you inject oligomycin, the basal oxygen level goes down, and then once you give FCCP, the maximal respiration takes place or maximum oxygen consumption takes place, and once you give rotenone or antimycin, again you have the decrease in the oxygen consumption. So this is how the oxygen consumption ratio measured in a Seahorse instrument. So next we will learn how we have to measure the extracellular acidification rate. So in the extracellular acidification rate, what happens is that the probe will measure the basal acidity of the medium, and after that the glucose is injected. So you know glucose is a substrate for glycolysis. Once glucose is injected, what will happen is there will be maximum amount of glycolysis that is taking place. So the acidity of the medium will increase further. So this is called glycolysis ratio. So once you give oligomycin, which is a drug that I already mentioned, it will prevent the oxidative phosphorylation. So once you give oligomycin, what will happen? Your oxidative phosphorylation is blocked. So glycolysis will increase further and it will increase to the maximum. So after that you block the glycolysis by giving an analog of glucose. So this analog of glucose is called 2-deoxyglucose. What it does is it will block the hexokinase enzyme, the activity of the hexokinase enzyme. So glycolysis is completely blocked. So your acidification rate will fall down to the minimum. So that is what is given in this graph. So these are the two measurements that we do to analyze the metabolism that is happening within a cell. So this instrument helps us to measure both glycolysis as well as oxidative phosphorylation without disrupting the cellular homology or cell physiology. So this is the advantage of this system, and with time you inject different chemicals. So how it is done? So this instrument has different probes. Basically it has two probes. These probes are injected within the medium that is in a 96 well plate. So you grow the cells in a 96 well plate and the cells are attached to the surface of the plate, okay, and you add the medium and the cells are happily growing. So once you inject the probe, once you keep the plate under the machine, there is a probe that is entering inside this media. So they have two sensors. This probe has two sensors. One sensor measures the acidity of the medium, the second sensor will measure the oxygen consumed, okay. So this happens by a phenomenon which is called photodiode, okay. So light is excited into the media and there is a photodiode sensor that is present within the probe. So this photodiode is what is measured, and that is what is measuring both the oxygen consumption as well as the acidity. Clear? So now we will see how we can utilize this instrument and measure the glycolysis in cells. So here we have given the examples of two different melanoma cells. So why we have taken melanoma is that it is a skin cancer and it is the most aggressive form of cancer that we can see. So once a melanoma comes, the patient will die within a matter of 2 or 3 months. So that is the maximal survival rate. So we have taken two cell lines of melanoma, MEL 526 and MEL 103. So you can see they have been treated with different concentrations of Lapacol. So 5 micromolar, 10 micromolar, 20 as well as 40 micromolar. So here what we have plotted as a graph is the basal as well as the maximal ECAR or the extracellular acidification rate that is measured in the Seahorse instrument. So as you can see, when you increase the dose of Lapacol that is treated, there is a gradual decrease in the extracellular acidification rate. So from this what we can see is that the Lapacol, which is the drug that we gave, is able to block the glycolysis and in a dose dependent manner. That is, when you increase the dose of the Lapacol, there is a decrease in the extracellular acidification rate. So that is the first measure of inhibition of glycolysis. Next, so let us think in this way. So once you block glycolysis, you have to see an increase in oxidative phosphorylation, right? So that is what we observed with Lapacol also. So when you measure the OCAR or the oxygen consumption rate, what you can see is that in both cell lines, MEL 526 as well as MEL 103, there is an increase in oxygen consumption rate with a dose. So that is 5, 10, 20 and 40, there is the increase in oxygen consumption rate. So that means your oxidative phosphorylation is increased. So whether it is because of the Lapacol and its direct action on PKM2, that was the next question, right? So in order to do this, we have the structure of PKM2 that has been plotted, and this is obtained from the protein data bank, and the structure that we used for docking is 1ZJH, okay. So this structure of PKM2 was taken and Lapacol, which is the drug of interest, was docked onto this one. This molecule, and as a control we used chiconin. Chiconin is another molecule that has been previously shown to inhibit PKM2, okay. So from this data, from the docking data, what we observed is that the PKM2 was inhibited at a binding concentration of 1.42 micromolar in the case of chiconin, whereas for the Lapacol, which is the new molecule that we have used, it was 141.86 nanomolar. So very low concentration of our drug was able to inhibit the activity of PKM2. So this is from the in silico analysis, that is computer based analysis. So whether it is true or not, we wanted to see this with the real enzyme activity. We have done in silico analysis. So computers can predict different things. So in order to see if in the real world the enzyme activity is inhibited by Lapacol, we isolated the enzyme from E. coli cells. So what we did is we expressed the PKM2 enzyme that is present in humans in a bacterium, that is E. coli, and then we isolated and purified this particular enzyme and we compared it with PKM1 enzyme. Okay. So we treated the enzyme that is purified with Lapacol and also the PKM1 enzyme with Lapacol, and we wanted to see the inhibition of enzyme activity. So from this graph what you have seen is Lapacol at different concentration. We used 5 micromolar Lapacol as well as 10 micromolar Lapacol along with the purified enzyme. So from the graph what we could observe is that PKM1 enzyme, that is the tetrameric enzyme, that is the normal enzyme that is present in normal cells, it was not inhibited. The enzyme activity was not inhibited by Lapacol at 5 micromolar and 10 micromolar, whereas the abnormal enzyme, that is PKM2, was inhibited at 5 micromolar as well as 10 micromolar, and the inhibition was increasing with the increased dose of the Lapacol. So that means we have a molecule that is specifically targeting only the mutant isoform of the PKM2 and not targeting the PKM1 enzyme. So this molecule can be used safely in a person because in his body this enzyme, this molecule Lapacol will not inhibit his normal enzyme activity, the PKM1 activity, whereas it will target only the cancer cells, because only the cancer cells I already mentioned have the PKM2 enzyme. So we have a novel molecule here. And then we also took the extract of PKM, that is pyruvate kinase activity, we measured in the melanoma cells. So we took melanoma cells from culture, we isolated the fraction, protein fraction from these cells, and then we did the assay. PKM assay was done in these fractions also, and in these fractions also we could see a dose dependent decrease with treatment of Lapacol with the PKM activity. That means in melanoma cells the active form is the PKM2 and not the PKM1. So here we could prove, so the cell that was used is MEL103, the melanoma cell line MEL103 was used for this particular study, and we could see that the pyruvate kinase activity of this extract was also inhibited by Lapacol. So that means we have the Lapacol as a new molecule. So if we treat the melanoma cells with Lapacol, if it is targeting the PKM2, what we should see is there should be a decrease in the division. For cancer cells, we expect the cancer cells to divide very fast. Okay, the normal division for a normal cell will be 24 hours, but whereas a cancer cell will divide every 2 to 3 hours. So whether we wanted to see if Lapacol treatment will decrease the cell proliferation rate or the cell division rate in the melanoma cell lines. Yes, in MEL526 as well as MEL103, we could see that with the increase in dose, the proliferation of cancer cells was drastically reduced. So this also helps us to study that Lapacol is a good molecule that will prevent the cell division in cancer cells. Then since Lapacol is able to inhibit glycolysis, and it is also able to reduce the, it should be able to reduce the ATP levels, overall ATP levels. So when we looked at the overall ATP levels in MEL526 as well as MEL103 cell lines, with Lapacol treatment the overall ATP levels of these cell lines also came down. So that means Lapacol is a molecule that can bring down your total ATP levels in a cancer cell also. So that means the cancer cells will have less energy to perform its physiological activities. So that is how the drug will target the cancer cells. Next, what we did is whether the Lapacol can be used to induce cell death in cancer cells. So now what we have done is Lapacol treatment alone, what it does, it will not help the cancer cells divide. So but we want to destroy the cancer cells, we want to kill the cancer cells. So in order to do that, what we did is we gave Lapacol along with another molecule which is called DNP or dinitrophenol. So dinitrophenol is just like FCCP or CCCP that was used previously in the Seahorse assay, right? So it is a protonophore, and this protonophore will disrupt the electrochemical gradient, and it will disrupt the proton gradient that is present within the mitochondrial membrane. So once this proton gradient is broken, the cell becomes susceptible to drugs. So that is what we see when you give Lapacol along with the lower concentration of DNP, you see apoptosis or cell death. So here you can see the flow cytometric data And this is the picture of mitochondria. So we know living cells will have functioning mitochondria. Here what we have seen in this picture is a fluorescent image of mitochondria in cells. So what you see as red spots are mitochondria that are respiring actively. So this is because of a fluorescent dye that has been used, or it is called mito tracker. Mito tracker red is being used here. So this mito tracker red will fluoresce will tag the mitochondria, and mitochondria will appear red in color. So here when you give DNP alone, or when you give Lapacol alone, you can see that the mitochondria are intact. That is, DNP alone, given alone, or Lapacol given alone does not induce cell death in cancer cell lines. But when you give them together, when Lapacol is added along with dinitrophenol, you can see that the mitochondrial membrane integrity is completely lost. So that is given in the fourth panel. So here you don't see the living mitochondria. So this means that DNP and Lapacol together will be able to target the cancer cells specifically. So we have published this in a journal which is PLOS One, and this was reported in 2020. And I would like to acknowledge my postdoctoral mentor. I was given the UGC Raman postdoctoral fellowship, and this work was done under the mentorship of Dr. Sami Subbu Naidu, and as well as Hal Boxmeyer. He is no more. I would like to acknowledge them, and I would like to thank you for your patient listening. Thank you.